Here's part 8 on page 93, and they're giving us a little glance here at the DNA. Here's DNA as a chromosome, and so the idea here is that this is a highly condensed chromosome. They generally will not look this way except as they're about to be dealt with in replication situations. So in general, either mitosis or meiosis, you don't tend to see it this way. But nevertheless, they're using this as an example in which to actually pull the string, so to speak, of one of these chromosomes out. And notice that the 6-foot length is being fit into a microscopic organelle called a nucleus, right? We're still inside the nucleus when we're looking at this thing called chromatin. So the dark, how shall I say, whatever that color is, purple, that's the actual DNA sequence. That's the sequence of base pairs. And it's being wrapped around proteins. Here, the individual proteins are beginning to make themselves apparent. These are called histones. There's a number of histone proteins that go together, and then there's one histone H1 that actually locks it in place. And we sort of condense the chromosome that we're not using. And then there's a portion of it that's naked that we are using, and that we're going to actually follow the sequence with an RNA polymerase and make a copy of a gene. It's going to be a gene that tells us something about ribosomal RNA. It's going to tell us about tRNA. It's going to tell us about messenger RNA. The messenger RNA is going to be the sequence that forms the polypeptide like insulin, or a protein, a globular protein like growth hormone, or something like that. So that's the idea here. Polymerases are really big, enzymes are really big proteins. So the idea here is that we're going to get the DNA, or rather the messenger, to come out, and first of all, it's going to bind to a free ribosome, and that's the very first thing that happens. And if that protein is destined to be used within the cell, the entire project of forming that protein, the translation process, the peptide synthesis, occurs entirely on a free ribosome. If it is intended for export, then the ribosome will actually begin its polymerization of the amino acid by making a brief length of peptide, and then it's going to seed itself on the endoplasmic reticulum, because ultimately this is how we get places within the cell. So that's the idea here, is that we're going to move around the cell, and that's part of what's going on here. So we're going to go from the rough ER, and ultimately we're going to go to the Golgi apparatus. Notice that the Golgi apparatus, although the artist has put it in a different color, it's just more or less more rough ER, but it's arranged very specifically from the rest of the rough ER. So let's take a look here. They have a blow-up of this. And the idea is that we have the ribosome producing the polypeptide or protein, and that the protein then is collecting over here in this vesicle, and it's leaving the ER. Do you see that it's leaving? We used to call that, back in the 1980s, blubbing off, right, and blubbing on. Now we call it the trans and the cis surface here of movement, right? So it sort of like emerges from over here, travels a little distance, it nuzzles up to the membrane here, and then merges with the membrane, as it were, and releases the protein in here. And then we have that going on again and again. Sometimes there's channels that you can travel directly through. Sometimes there's modifications of the proteins that happen here, and then eventually we get to export vesicles that eventually merge with the cell membrane, and we release our exported molecules right here. So that would be our secretion. This would be the case if we're talking to other cells by way of insulin, or glucagon, or any other luteinizing hormone, and so on, follicle stimulating growth hormone. So we could be producing molecules which are to be released for use in other parts of the body. Okay, so that's the export mechanism. And then we have another type of thing that goes on in which we have, they're not showing it here, but there is a thing called smooth ER. And I just want to sort of like bring that to your attention a little bit. First of all, that this is not a place in which we make protein. This is a place where we do something entirely different. And they give us some bullets here. There's certain parts of the body that specialize in cells that have smooth ER, and that would be particularly in the liver. The liver has a lot of functions because all of the nutrients coming from the stomach, small and large intestines, all of the blood, the venous blood, is going to go to the liver by way of the portal vein. And so the liver turns out to be a pretty special place. And what does it do for a living? It metabolizes lipids, it synthesizes cholesterol, synthesizes lipid components. Okay, so one of the things that the liver does, they're not showing it here, are they? But whenever you have an adequate supply of nutrients and calories, you can change some of the glucose of your diet actually into glycogen and store it. And then you can also produce fat when you've actually stored adequate amounts of glycogen. So glycogen here, the storage is discussed in the last bullet. Here we're discussing if you have enough glycogen, you're going to make some fat or some lipid to go to the adipose tissue. Synthesis of steroid-based hormones, sex hormones, and so on. That's something that happens, and that would require you to be in the right place for that. So the smooth ER of gonads would be where that happens, or wherever steroids are made. It could be the adrenal medulla, the adrenal cortex, not the medulla. We have absorption and synthesis and transport of fats in general. If there's fats in the diet, you can actually deliver a fat with only about 3% loss of calories directly to the fat. So we're really good at sparing, if we don't see food for a while and you have something that's fatty in the diet, you're going to actually store all that fat, pretty much 97%. We detoxify drugs, and they talk here about pesticides, which at any rate, basically any sort of toxins. This is something characteristic of the liver as well as the kidney. The liver and the kidney both metabolize drugs. If there's a drug therapy going on that's significant, like chemotherapy, the liver and the kidneys actually often become enlarged because of their need to do so much detoxification. Okay, and then as previously noted, we break down or form glycogen. Okay, we can break down glycogen to form free glucose, or we can actually store. And we don't have anything else discussed up here at this level. Okay. One of the things that they're noting here is that we can actually store glycogen in skeletal muscle, cardiac muscle, and smooth muscle. So that's another storage site. Okay, so the only thing that we have left to talk about now is the mitochondria, and we'll pick that up next.